we're an intratumoral immunotherapy company, arming the immune system to fight cancer. Here's our forward-looking statements, which I'll give you a moment to peruse. That should be enough time. So, OncoSec's platform solution. So what's the problem that this is a solution to? Fundamentally, it's represented by this cartoon that I'm about to show you, which is that tumor cells deploy multiple parallel mechanisms to subvert the immune response. And it's our hypothesis that we need to push back in a similar way by deploying multiple parallel mechanisms to revitalize the immune response in the tumors. This is depicted here in this cartoon by, positive, or what people call the gas pedals or immune drivers in green, and the negative regulators of tumor immunity in red. And basically, in a nutshell, what we're trying to do is to change the picture of a tumor from red, meaning a locus of immunosuppression, into what I will tell lay people is converting the tumor into a lymph node. Now, that's not technically true, but I think functionally, it's really what we're after is to re-hijack the immune system in the tumor and drive immunogenicity. Now, those were just a couple of different players. Of course, if you really want to think about that, it's probably got scores of pieces and more like a Rubik's Cube than that puzzle there. So OncoSec, why are we different? Well, we're an intratumoral immunotherapy company that is both a device and a drug. We have a technology in intratumoral electroporation that allows us to deploy many different genes. So it's intratumoral gene therapy. And unlike oncolytic viruses or other platforms, we're really not restricted in terms of the number of genes that we can put in, or less limited to by the packaging size of, say, a virus. So currently in preclinical studies, we're putting in multiple genes on one plasmid, up to 5 or 6 at this point. What that allows us to do is to tackle multiple facets of that immunosuppressive microenvironment. Company strengths, we already have demonstrated proof of concept in several completed phase 2 studies in Merkel cell and in melanoma. And it's a rapid preclinical and even getting into the clinical proof of concept stage because our drug is DNA. And so we have a much faster and cheaper CMC and tox path in that regard. So we're publicly listed on the NASDAQ, and we have cash runway through Q1 2017 to achieve some of the milestones that I'm going to talk about here. I'm going to go to this slide first because it really, I think, presages the other one, which is, how does this work? Quite simply, you inject your payload here. Our first molecule is interleukin 12. You inject the DNA-containing solution into the tumor. You follow it with brief electric pulses that permeabilize the cell membranes. The DNA goes into the cells. It then gets expressed. In the case of IL-12, it then gets secreted into the tumor microenvironment, and it generates a local inflammatory setting. IL-12 is a prime driver of the Th1 side of the immune response, which we know is really the most effective type of immune response against cancer. So in short, we're reprogramming the tumor and teaching the lymphocytes to see antigens from the tumor, in particular Neo-Antigens that are patient-specific. So going back to that slide. So we train the immune system to target and attack cancer. It's based on a local delivery, but it's not just a local therapy. Our whole goal is to get an abscopal effect, a systemic immune response driven by a local therapy. We could put together lots of different molecules together in a synergistic, orchestrated manner. Again, we are this technology, by leading to tumor cell death in immunogenic fashion, releases all the potential antigens in those cancers. So you don't have to a priori pick an antigen of choice, but you expose the immune system to all of those potential antigens and let the immune system sort it out. And that, of course, gets to those potent Neo-Antigens, those non-synonymous somatic mutations that encode for protein alterations that the immune system sees as non-self. So preclinical and clinical proof of principle here. When I was at Merck working on the PD-1 program, I initiated some of this work that was later published together with Tony Ribas and Paul Tomei in this Nature article, where we really defined what the responder and non-responder phenotype in melanoma looked like. And since that time, it's been extended to many different types of solid tumors. In a nutshell, and to a first approximation, the patients who don't respond to PD-1 or PD-L1 drugs are those patients where their tumors are not immunogenic. They don't see the tumor as foreign. They don't send in CD8 T cells. And so there's no target cell here to treat with pembrolizumab. On the other hand, patients that look like this, where they have the ability to see that tumor as foreign, they mount the CD8 defense, the CD8s go in there, they make interferon gamma, that upregulates PD-L1, and so you get this immunologic stalemate occurring, which we have termed adaptive resistance. These are the patients who respond to pembrolizumab and other checkpoints. The patients, however, the bulk of patients look like this, where they're poorly immunogenic, they're not generating that immune response. This unmet medical need is huge. This is just an example of what the sort of numbers associated with some of these solid cancers are. In general, the PD-1 or PD-L1 checkpoints have response ranges in the range of 20 to 40%. So there's a huge unmet medical need there. Now, we tried to OncoSec grew out of a successful phase 1 experiment before the company even existed, and then when we were founded around that phase 2 asset in melanoma, and we didn't really have much of the biology underpinning what we were seeing in the clinic. So over the last year and a half, we've built an R&D group to really try to understand how IL-12 was working and how it was driving a systemic immune response. What we did was model what we were doing in the clinic here with B16 melanoma cells, three treatments in a week. This is their clinical paradigm. You see that we're extremely good at curing the treated mouse tumor. This is interesting, but not essential. What we're really after is a systemic immune response. So we built a mouse model with 2 tumors and 1 on either flank. We treat one, leave one untreated. And you can see here we have a significant reduction in the size of that untreated tumor. What's really interesting here, however, if you look at the individual animals, they behave a lot like we see in patients, where some patients just continue to progress right through the therapy as if nothing has happened. Others stabilize, others go on to a response, still others go on to a complete clinical, mouse clinical response. And in our studies, this range of CRs ranges from about 10 to 40%. So we want to understand what is unique about those mice that respond so well. So we began to look at what we were doing in terms of driving tumor-specific antigen CD8 responses. And you see here, we use a test antigen, Synfecal, which is derived from ova in the B16 ova model. And we do that treatment, we see we're generating a huge amount of Neo-Antigen specific CD8s, and that corresponds with efficacy in this preclinical model. Interestingly, in people, this was discussed at the recent ESMO meeting in Vienna. This is a patient from our Merkel cell trial who had undergone multiple therapies, including chemotherapy, radiation therapy, interlesional therapy with an interferon, I believe it was a beta, and really didn't respond at all. After treatment, however, with IL-12 intratumorally, the patient began to have a response, and this response evolved over months, and we could pick up using a tetramer assay that's specific for the polyoma virus Neo-Antigen in Merkel cell. We could see this evolution of this tight cluster of polyoma-specific CD8, showing that also in a patient, the IL-12 ImmunoPulse therapy is acting as an in situ vaccine and generating an increased tumor antigen-specific response. This is just a summary of our phase 2 melanoma data, where we had an overall 31% response rate by a skin-modified RECIST, a complete response rate of about 14%, disease control rate of 48%, but I think most importantly, in 50% of the patients who we had an untreated lesion that we could measure and follow, they had an objective regression showing a high frequency of the so-called abscopal effect, or systemic response to a local therapy. If you look at what's happening in these tumors, you see IL-12's ability to drive TILs. This is an example from a phase 1 patient who went on to get a complete response.
You see here, there's almost no signal. This is really a T cell cold tumor. After IL-12, however, it converts into a very T cell rich tumor, and this is a higher magnification, showing again this interdigitated PD-1 and PD-L1 expression that we know is a signature for response to pembrolizumab. In Merkel cell, we saw a similar phenomenon in several patients. Interestingly, in this case, there was a generation of necrosis and a significant infiltrate, but this patient went on to actually progress. And we feel that this is likely due to these T cells getting hung up on the PD-1 checkpoint, which makes the rationale for doing this combo study, I think really what I usually refer to it as the duh rationale, because it's just so straightforward. Here you take a tumor that doesn't have T cells, you use IL-12 to generate immunogenicity and to educate the immune system to see those antigens and drive a TIL response. Those T cells then traffic to disseminated lesions that you haven't treated, but you would predict that they would run into the PD-1, PD-L1 checkpoint, and that our monotherapy effect would be stunted by that checkpoint. So the hypothesis would be that if you were to block PD-1, that in fact you'd get synergy. And that's being tested currently in a combination trial together with pembrolizumab that is an IST through UCSF. So that's basically our lead program, which is in the clinic, which is IL-12 is the molecule we deliver. But there are lots of other molecules that are present in the tumor microenvironment that one may want to target. And as I said, I think the defining feature of our technology over and against others is the ability to stack multiple genes on top of one another. So there are many of them to choose from, and we're currently going through lead identification at this point to develop a combo therapy to even better drive the immunogenicity of the tumor. I'll just point out that IL-12 and interferon gamma probably has as high a monotherapy response rate as we've seen due to the fact that it intersects at multiple aspects of this tumor immunity cycle already. So what have we done so far? We have positive data from phase 2 clinical trials in melanoma and in Merkel cell. This is really a strong clinical proof of concept of this platform technology. We've shown that IL-12 drives tumor-specific CD8s and abscopal responses, and we know we can deliver multiple immunomodulatory genes in combination in our preclinical setting. In fact, IL-12 is a combination. If you know, it's a heterodimer of P35 and P40, so it already is 2 genes. And we think that this is an ideal combination partner for checkpoints. This is a slide I'm going to skip, which is these numbers are usually quite fanciful, but obviously there's a huge market opportunity in tackling that unmet medical need of poorly immunogenic tumors. We already covered this. We have several phase 2 trials going on, the combination with pembrolizumab. We have a pilot study in triple-negative breast cancer and in head and neck cancer. These are, I've already discussed the rationale for this. And really, triple-negative is a focused study where we're treating the tumors and identifying with pre- and post-treatment biopsies whether or not we're moving the immune system in the right direction and driving immunogenicity and a TIL response. We'll skip through that. Some of our preclinical collaborations, which we've talked about in the past, are a preclinical collaboration with Heat Biologics and their GP96 Ig molecule. We're working with Paul Tumay and PerkinElmer to develop those lovely multiplexed IHC images that you saw, and we're working with Plexicon's compound to see if we get a synergistic effect when we block CSF1R with their inhibitor. Now, in terms of clinical and commercialization strategies, we have really 2 basic arms: find a partner or do it ourselves. Our developmental milestones for this next year are really to announce a new key academic or industry collaboration, expand the proof of concept phase 2 melanoma combo study sites, and increase enrollment there, and generate really more data around the testing the hypothesis that PD-1 inhibition and IL-12 will synergize. We're going to move forward with this triple-negative biomarker study, as I discussed. to show that IL-12 is capable of doing this in harder to treat, not as immunologically sensitive kind of tumor as melanoma or Merkel cell. We're going to announce a multi-gene combination in this year, and we're going to present preclinical combination data with heat biologics. This is our team, and I think this is some of our financial stats. I don't even know what any of this means, so don't ask me questions about that. And thank you.